Hi everyone, welcome to the second video in the unit on adaptive immunity. In this video, we're going to talk about a concept called antigen presentation. This is how antigens that have entered the body get shown or presented to the T cells of the body in order to start the adaptive immune system response. So let's go ahead and take a look. Antigen presentation is the process of presenting foreign antigen between dendritic cells or macrophages, those antigen-presenting cells of the innate system, to T cells of the adaptive system. And so to understand antigen presentation, you have to have a general sense of what a T cell is. The T cells are going to be controlling the cell-mediated responses of the adaptive immune system. These are the responses where it's the cells themselves that are off doing something. Remember, they all mature in the thymus, but there's different categories of T cells, and they're all distinguished by what we call surface proteins that are on the cell membrane itself. You'll see as we look at the immune system, there's often names called CD with a number. So you can see here CD4 or CD3. All those are is designations of immune proteins, and we use them to differentiate one cell type from another. So for the T cells, there's really two categories that we're going to look at for this class. You've got CD4 cells, which you can see here. Here's that CD4 protein that would be sticking off the surface of that type of T cell. There's a different class of T cells that are called CD8, which would have a different protein here in place. You're going to want to remember that CD4 receptors are going to interact with MHC2 molecules, and CD8s are going to interact with MHC1. CD4s are also called helper T cells, while CD8s are called cytotoxic T cells. And a lot of those words might not mean a whole lot right now, but you'll understand them better as we walk through these processes. All T cells, no matter what subtype they are, all have a T cell receptor, which is what's shown here. So we abbreviate that TCR for T cell receptor. This is what actually binds to the antigen. So these are the kind of little shapes that I had been drawing when I was drawing the cells before that had these little receptors with this different shape. This is the antigen binding site of this shape. And so it's here that we would say have that little circular antigen that could be bound. And then the other molecule that we see in here are these CD3 proteins. These are needed for cell signaling, so that process of clonal selection. Remember, when one T cell binds the antigen, it then activates to undergo rapid mitosis. That activation is done by those CD3 proteins. The key here is going to be the way that I've drawn this, with the T cell receptor just kind of binding this antigen, isn't how this actually works in real life. T cells can't bind antigen that's just floating around in the cell like this. So this binding actually wouldn't happen. It has to be presented to them by those antigen presenting cells on proteins that we call MHC molecules. And that's what we're going to look at for the rest of this video. Okay, so remember the simplified version of a T cell with a T cell receptor that was going to bind to an antigen. This is just a simplified version and isn't really how the process works. Instead, what's happening is something called antigen presentation, where you have a cell, typically a dendritic cell, that is going to present the antigen on what's called an MHC molecule. And there's two types of MHC, MHC1 and 2. We'll talk about the difference in a minute. So instead, the dendritic cell holds the antigen out here on the MHC molecule, and the T cell, with its T cell receptor, is able to interact with that MHC. This is called antigen presentation. And it's the antigen presentation that's going to then activate that clonal selection for this specific lymphocyte of T cell that's being presented to. There's two ways antigen presentation can happen. Either presenting of proteins that were in the cytoplasm of the presenting cell, or in presenting antigen that had been enclosed in a phagosome that had been taken in through phagocytosis. Those two pathways are what the difference between our MHC1 or our MHC2 molecules are. Okay, so MHC stands for major histocompatibility complex. Remember, these are the surface proteins we just mentioned that are found on surfaces of cells that can present antigen held out here on their surface to those T cells. Your MHC molecules are actually highly variable, so your MHC proteins that are presenting antigen would look different than someone sitting next to you. And the reason they're called major histocompatibility complexes is because these are actually what we need to match when we do organ donation. So when you hear about people trying to match tissue types, that's really what they're trying to do, is match those histocompatibility proteins so that they are the most similar. So we said there's two types. We've got MHC1 proteins, and you can see the structure of an MHC1 here, and we've got MHC2 proteins. You can see the structure of that here. Structure is a little bit different, but that's not really the key piece of the main differences between them. So MHC1 proteins are going to be found on any of your nucleated body cells. So just about all of the cells in the body have MHC1 on their surface, whereas MHC2 is specialized to only macrophages, dendritic cells, so those two antigen-presenting cells we've talked about, and B cells. When MHC1 presents antigen, so when it puts antigen out on its surface like this, it's going to interact with CD8, which are also called cytotoxic T cells. As opposed to MHC2 molecules, when they're presenting antigen, they're going to present antigen to the CD4 or the helper T cells. And that matters because the type of antigen they are presenting is different. So MHC1 is presenting intracellular antigen. This is proteins that the cell itself was making from its own ribosomes in its cytoplasm, which means these intracellular antigens can become problematic if you have things like a virally infected cell, or if you have cancerous cells where their proteins are not forming properly anymore. That's what MHC1 and those cytotoxic T cells are supposed to be focusing on. MHC2 is used for extracellular antigen. This is antigen that's been phagocytosed. So that's, remember, macrophages and dendritic cells can do phagocytosis. So this extracellular antigen has been brought into the cell, into a phagosome, and is then presented out on the surface. So this is typically used then for things that are outside the cells, so bacterial infections or viruses before they've actually gotten inside a cell. We're going to look at how the presentation happens for both of these types. Okay, so for presentation of cytoplasmic proteins, remember here, this is going to be a virally infected cell, so the virus is getting in. Remember, it's going to have to uncoat. We start transcribing and translating viral proteins. But in the cytoplasm of all your cells are these structures called proteasomes. The proteasomes are there to digest proteins in the cytoplasm. And they are normally digesting all of your self proteins, because your proteins have to be recycled. They get old, we break them down, and we reuse the amino acids to make new proteins. So this proteasome is digesting both self proteins that are here in the cytoplasm, along with the viral proteins. Some of those pieces end up in the endoplasmic reticulum. And if you remember, the endoplasmic reticulum is part of the endomembrane system. To get things to the surface of the cell, they have to enter that endomembrane system here at the endoplasmic reticulum. Once the peptides enter, so those little pieces of digested viral protein, they get loaded onto the MHC1 molecule, and then it gets packaged in a vesicle, goes to the Golgi, goes into another vesicle, until eventually it makes its way to the cell surface, where now we have that MHC1 molecule with its specific antigen here. So remember, this is the antigen. And it's now going to interact with the T cell receptor of this T cell. And notice the co-receptor. So this is the CD8 co-receptor. That tells you that this is a cytotoxic T cell. And that cytotoxic T cell, we'll look at the actual functions of them in the next video. But once this interaction happens, now the cell is activated and it's ready to go do its job. But remember, we had to have the MHC1 molecule presenting to the cytotoxic T cell in order to have this activation happen. and that occurs with these cytoplasmic proteins. We'll now look at a similar process of presentation of phagocytized proteins. So instead here, remember the proteins were originally extracellular, so they were out here outside the cell, and these could be bacteria that were being taken in by, say, a dendritic cell. They could be viral particles, or they could be pieces of pathogens that maybe were killed by neutrophils during inflammation, and now the dendritic cell is just pulling in the pieces. But either way, they were out here outside of the cell. And so we call those exogenous antigens, things that were found outside the cell itself. And they're brought in through phagocytosis. And remember, the phagosome fuses with the lysosome to make our phagolysosome, which is then chopped up all of our proteins into these little individual antigens. The phagolysosome is now going to fuse with a vesicle that's coming from the endoplasmic reticulum. And what's in that vesicle is this MHC2 molecule. So remember, the MHC1s were used on the last slide. Now we're looking at MHC2s. This vesicle with the MHC2 will fuse with the phagolysosome, and when it does, it will get loaded with the antigen. This is then displayed out on the surface. So here we have our MHC2 molecule with its antigen, and it's interacting with the T cell receptor of this cell, remember the one that matched the shape. And notice here, this co-receptor is now a CD4. That tells us that this is your helper T cell that is now going to be activated by this reaction. So remember, helper T cells are acted by exogenous antigen presented on MHC2 molecules.